It's great to see everybody. Good to have you all here. Our speaker today is Serena Sanulli. She is a Chan Zuckerberg BioHub investigator at Stanford. And it's been wonderful to watch, her career evolve. Serena loves everything about chromatin, dynamic, structure, function. She studies nucleosomes and chromosomes with a wide range of biophysical, biochemical methods. And naturally, no surprise to anybody on this Zoom meeting, that research interest has led her to condensates, quelle surprise. And so today she'll be talking with us about her work on HP1 and how that drives chromatin compaction into heterochromatin. And Serena, as I mentioned, has worked in chromatin her entire career. She got her PhD in 2013 from the Curie in Paris, studying chromatin changes and development with Raphael Margaron. And then she moved to UCSF and she worked with Gita Narlikar and John Gross, and that was to apply biophysical methods to study chromatin structure and organization. And just a few months ago, she took her faculty position at Stanford, and she's just getting her lab up and running, of course, in difficult times, but getting off to a great start. And so we're also very grateful to her for her willingness to give a lecture to all of us so early in the morning out there in California. So it's clear that she loves the condensate community just as much as she loves chromatin. So Serena, the floor is yours.
Thanks, Mark. Thank you so much for the kind introduction. And it's really a pleasure to be here with you, virtually with you today. It's also being the part of this condensate community and line up with the series, this kitchen talk series that you've been putting together. It's great. So today I'm going to tell you about chromatin and my long-standing interest, and also going to tell you the journey within the phase separation field as a chromatin biologist. And so I have to confess that I didn't really appreciate the implication of phase separation from the beginning as a chromatin person. But what I'm trying to do today is really going to tell you how the perspective of phase separation has really reshaped my view of chromatin organization. I'm going to try to really share with you this vision. Okay, so can I move on? Yes. So in eukaryotic cells, the genome is packaged into chromatin and is stored in the nucleus. And here in this beautiful image, you just see the nucleus of a eukaryotic cell, and you can appreciate the complexity of this environment and the presence of this subcompartment or subdomains inside the nucleus. So some of these are very large subcompartments. You see the nucleolus here. Some are smaller, such as, for example, here, the PCG bodies. So these different compartments are different for their composition, but also for their function. And recent work has shown that a lot of these compartments are they're highly dynamic, and they form through mechanisms that involve a phase separation. What I'm interested in is trying to understand how the genome and chromatin organized inside this complex environment. So if you look at the nucleus of a cell, and this, what you see here is a section of a nucleus seen by electron microscopy, we can immediately see the chromatin inside the nucleus, and we can appreciate the heterogeneous structure of chromatin. And we can see that there are dark patches shown here, darkly stained, and then light patches of chromatin. And based on this cytological appearance, chromatin has been divided into heterochromatin and euchromatin. So the heterochromatin are these dark regions, are considered the most dense and packed region of chromatin, and they have been associated with low transcription. The light regions here, the region that are called euchromatin, are considered to be the relaxed and open region of chromatin and associated with active transcription. So I'm really interested in understanding what's the structure of chromatin inside these different patches, what's the difference, and how can chromatin structure, these subdomains, be regulated in a dynamic way during the life of a cell. So to highlight, and the reason just in terms of chromatin organization, it's because we realize that the way the genome gets packaged in this chromatin, it really regulates almost every single process of DNA, including transcription and DNA replication. So to highlight the different possibility and types of chromatin organizations that we can see in the cell, I'm just showing you different cell types by electron microscopy. Up here, you can see the hematopoietic stem cell. These are hematopoietic stem cell, and these are different cell type from hematopoietic lineage. And so if you start to see inside the nuclei, you can see that the dark and light patches are actually very different across different cell types. So I tend to think of this as chromatin mesoscale organization, how chromatin is organized and fold in 3D. So the ratio between heterochromatin, euchromatin, dark and light region is actually very different. But then you can also see the respective nuclear positioning of this different type of chromatin is also very different. So this is just to highlight how dramatic difference chromatin can have in terms of organization in the cell, and also highlight that this chromatin regulation is really important to specify cell function. So what do we know in terms of chromatin organization? I always like to show this hierarchical textbook model of chromatin organization. Probably most of you have seen it. In the left side of the slide here, you see the cartoon of this model. And on the right side, I'm showing the corresponding micrograph from electron microscopy. So this is considered a hierarchical model because it goes from the naked DNA down here up to the most condensed type of chromatin that we find in the mitotic chromosomes. And so I'm really interested in looking at the first level of packaging, of DNA packaging to the chromatin down here. And today I'm going to talk a lot about this. They're really the first level of packaging of chromatin. When we think of packaging, we think of nucleosomes. And so basically it's when the DNA gets wrapped around a group of protein, which are called histones, to form this fundamental basic unit, which are called nucleosomes. And you can see here, if you look in the electron micrograph, if we look at an open stretched out fiber of nucleosome, they have this appearance that has been called as beads on a string, where the nucleosome, the spheres are beads and the connecting DNA is a string. So the next level of compaction is in this model, what you see here is this fiber, which is called a 30 nanometer fiber. And this fiber is formed by a chain of nucleosome folded in a very specific way, creating specific nucleosome-nucleosome interactions. And this was thought to be a very static, discrete kind of structure that has a diameter of a 30 nanometer fiber. And you can see here how this fiber looks like. When you extract chromatin from the cell, you treat the cell and you treat the chromatin, you put that on a electron microscopy grid. So, of course, things are not so simple. And so we really have a relatively good understanding of the atomic details of the nucleosome itself. So they're really the fundamental unit of chromatin, the building block of chromatin. So here I'm showing the high resolution crystal structure that we have available today. And so a lot of work has been focused in the chromatin field in studying the tail of the histone, which you see here, they're protruding out of the nucleosome core. So these tails are considered to be highly flexible. They can be modified by different type of post-translational modifications. And a lot of work in the chromatin field has been focusing on these PTMs because they seem to regulate dramatically chromatin. But for some reason, the chromatin people assume that the folded core, this central part of the nucleosome, was a rigid unit. And actually, if you look at the initial textbook model of chromatin, they refer to the nucleosome core, the histone core, as rigid Lego block coming together to form this very static structure. But in the most recent work in the last couple of years in the chromatin field, we have started to appreciate that actually there is something going on also in this folded core of the nucleosome, and then we can see some structural flexibility in there. And I'm gonna talk a lot about this today. And one interesting aspect for in the future, an aspect I'm really interested about and we focus on in my lab, is understanding what is the extent of the conformational plasticity in the folded core of the nucleosome and what's the function of this. When I started to look at what's going on in terms of chromatin mesoscale organization, how chain of nucleosome come together and form 3D structure in space, I was really surprised to see that what we know is actually much less. And that the in vivo data have failed to basically reproduce and show us what we've been able to see when we extract chromatin from the cell and put that on a grid. So to give you an example, when we used to think about euchromatin and open chromatin structure, we are used to think of nucleosome, like it's shown here, far apart from each other, with the idea that transcriptional machinery could, for example, access the underlying DNA. On the contrary, when we think about dense and packed packages, we have been thinking of a structure that could look like the 30 nanometer fiber, so very compact and dense type of chromatin. The problem has been that when we have started to look in vivo into cell, we really fail to observe the 30 nanometer fiber or any kind of discrete defined structure. And if we look at the most recent data based on imaging, and this is an example from a most recent technique based on electron microscopy, but also we think about 3D chromatin conformation studies, all these data are pointing out at the fact that in cells, chromatin does not adopt any highly ordered kind of structure. And the nucleosome seems to adopt, the chain of nucleosomes seems to adopt a relatively disordered kind of structure. They form clusters that can go from 5 to 25 nanometers. And you can see up here, you can really adapt many different kind of conformation. And the exciting part is that if you compare the dark patches versus the light patches, so heterochromatin versus euchromatin, we don't see any specific difference in terms of structure. And the only difference is the fact, basically the density of chromatin, how many nucleosomes we have in a defined volume. And so really understanding what is this, is there a specific structure for heterochromatin or is there a specific structure for euchromatin, I think it's been a quest for the chromatin field. So if we start to think about chromatin in the cell and chromatin organized through not a defined structure, but more kind of a disorganized structure, to me is really puzzling to conceive how cell can set up highly regulated processes starting from a chromatin structure that is disorganized and messy. So is it really that chromatin in the cell is like a spaghetti ball with no any kind of organization? To kind of understand better the context of chromatin compaction, I used as a model system the HP1 type of heterochromatin. So this is a highly conserved type of chromatin. This is the reason I decided to choose this as a model system. It's conserved and it's present from yeast to human. And when I started working on HP1 heterochromatin, we also had a fairly good understanding of the molecular player involved in this process. So we know that the key component of this type of compacted chromatin is the HP1 type, the HP1 protein shown here in green. So these are structural protein. They are known to specifically bind chromatin containing as a post-translational modification, the methylation of lysine 9 of histone H3. HP1s are essential for the formation and the function of heterochromatin. So we know that the way in which they work, the model that has been proposed in the way HP1 protein work is that they compact chromatin and therefore they induce gene silencing. And I always like to share this video that I think very visually show how good HP1 proteins are in compacting DNA. This is a DNA curtains experiment. The DNA is fluorescently labeled. And then when you add HP1 protein, you can see that the DNA gets packed up towards the top of these DNA curtains. And so HP1 proteins are really good in compacting DNA. And the question that I had is, how is this is happening at the molecular level? So we had some good understanding of how this protein works. And one of the models that have been proposed has been called the nucleosome bridging model. And this model relied on the fact that HP1 proteins can actually bridge across nucleosomes. So biochemical work had showed that HP1 proteins can form oligomers. And so they can form really this long chain of HP1 proteins that also can bind chromatin. And so this led to a model in which the formation of this oligomer could mediate bridging across nucleosome chromatin fiber. And then more recently, what has been shown is that HP1 protein also can phase separate and form this condensate, these droplets. And you can see this in vitro. This is a recombinant HP1 protein, human HP1 protein alone in a test tube forming condensates. And you can see in vivo, this is GFP tag HP1 alpha in Drosophila. HP1 formed these very bright foci that are highly dynamic. They fuse and they come together. And so the behavior of HP1 that has been observed in vivo is really consistent with the liquid-like property that we see in the test tube. So the observation of HP1 phase separation really raised a lot of excitement, and especially in the chromatin field, because this really led to a very simple intuitive model to think about chromatin compaction. So this is a cartoon of the model, and this model, the idea is that HP1 proteins can accumulate on chromatin, and then once they reach a certain critical concentration, they will phase separate. And then chromatin, which has high affinity for HP1 proteins, will be sequestered, dragged inside this condensate. And so that could be a mechanism that could be implicated in gene repression and compaction. But actually, how and whether HP1 phase separation is related to chromatin compaction was still an open question. And for me, that really meant trying to understand what was the role of chromatin inside this condensate and how HP1 proteins interact with chromatin. So I don't think I need to tell this audience about phase separation and remind that the formation of this mesoscale structure, these mesoscale droplets, is actually driven by molecular interactions. And these are interactions between molecules, but specifically multivalent interactions that are weak and transient. And so if we think about HP1 proteins, and what we knew from previous biochemical work is that HP1 protein can oligomerize, so they provide multivalency, and so and we know that this oligomerization is actually essential for phase separation. If you break it, you also prevent phase separation. So this made sense, and so the question is still open, and I think the question I've been mainly interested in trying to understand what's going on with the chromatin. What's the role of chromatin when it's dragged inside this HP1 condensate? To understand HP1 chromatin interaction, I decided to start with the simplest model possible for heterochromatin. So I use one nucleosome that is shown here in this cartoon in association with HP1 protein. And I use the simplified system to really try to understand the complex, but by using a combination of in solution methods. And I used a hydrogen deuterium exchange coupled to mass spec, and I refer to this as HDXMS. I use NMR, and then I use cross-linking mass spec. And the reason I choose in solution method is that because I really wanted to learn about the conformational changes and dynamics of the complex in solution and know what was going on also on the nucleosome itself. But also this was a kind of a choice that was driven by the fact that the conventional structural methods such as cryo-EM and and crystallography had failed to solve high resolution structure. And this was a real indication that this was a highly dynamic complex. And we also knew that a large portion of HP1 protein was formed by disordered protein regions, making it a very difficult target for structural studies. I'm going to spend some time now to describe the HDXMX experiment so you can start to understand how this technique works, but also the kind of information it can give us in terms of conformational changes of a complex. So in the experiment that I did, I incubated a nucleosome alone or a nucleosome in complex with HP1 in a solution that contained deuterium. So what happened is that over time, the hydrogen of the backbone of proteins can exchange with the deuterium in solution, and we can monitor this deuterium uptake by mass spectroscopy. So basically, you can think of this deuterium uptake as a proxy of solvent accessibility. So you would expect that regions that are on the surface of the nucleosome will exchange faster than regions that are buried into the core. So those will be regions that will show faster deuterium uptake because they're more exposed to the solvent. But you would also predict that the binding of a protein such as HP1 on top of the nucleosome would cause a reduction in deuterium uptake. So it's lower deuterium uptake at the region of the interface. And so especially when I performed this experiment, I was really expecting to see some level of protection because of HP1 protein binding the nucleosome, but also because of the idea that we add of HP1 protein as a group of protein that create a more compact and less accessible chromatin state. But actually what we saw was exactly the contrary. So to guide you through this slide, on the top here, you see the 4 different histones, and with their secondary structure mapped here. And then all these different bars are peptide that were sequenced by mass spec. And the color code is telling us how much the deuterium uptake is taken up in the nucleosome bound state. So basis the differential deuterium uptake of the free versus bound nucleosome state. We can take all these peptides and then map them on the structure of the nucleosome. And what we immediately start to see is that the increase in the deuterium uptake is really widespread across the whole nucleosome. And these mean that all this blue region that you see in the crystal structure here becomes more exposed, more accessible to the solvent when HP1 is bound. And I say this was pretty surprising, given the idea that we had of HP1 protein as a group of protein that mediate chromatin compaction. I also want to show you some specific region of this nucleosome to highlight how dramatic this exposure was. Those are regions that are now highlighted in orange and red. Those are regions that becomes dramatically exposed when HP1 is bound. And if you look now on the surface representation, you can see that those are regions really buried inside the folded core of the nucleosome. And those are really hydrophobic regions. So this meant that the HP1 proteins are binding on top of the nucleosome, and they're really opening it up, exposing all these hydrophobic regions. So this was very surprising data, and I was glad that I had complementary methods that I mentioned before, cross-linking mass spec and NMR, to support this major rearrangement observed in the nucleosome structure. So just to briefly tell you about the NMR experiment, I'm not going to go into the detail, but actually NMR is a very powerful technique that can give us information at the atomic scale resolutions of residue level resolution about motions. Conformational changes in that specific residues. And what I was able to see with the NMR experiment is that some of these residues, which are shown here as red sphere, undergo major dynamics as a consequence of HP1 binding. And those are, again, are residues buried inside the nucleosome core, indicating that, again, HP1 is binding on the top of the nucleosome and inducing motion inside the core. And so this observation and also the kind of changes that we observed on the NMR data indicated that there were dynamics on the millisecond, microsecond time scale occurring within these specific residues inside the nucleosome core. And this data really suggested that there was a conformational change occurring in the folded core of the nucleosome. So together, NMR, HDX, but also cross-linking mass spec that show major rearrangement in terms of cross-link context when the HP1 was bound, really indicated there was something major going on in the nucleosome core, and that the binding of HP1 opened up and reshaped the nucleosome structure. So that's how I start to think about nucleosome and the idea that there is a canonical state, which is likely what we see when we look at the crystal structure, but also the fact that the binding of proteins such as HP1 could increase the conformational dynamics of nucleosome, so they can sample conformations that are different from the one that we used to think about when we look at the crystal structure. So now the question, of course, is why this is happening and why HP1 is inducing such a conformational change in the nucleosome, and is there any function of this in the context of chromatin compaction? To look at chromatin compaction, I decided to use an assay, a self-association pelleting assay that has been used in the chromatin field for a long time. It's a very simple assay. You obtain chromatin used a chain of nucleosome of 12 nucleosome, and this fiber under normal conditions are soluble in solution. But if you add a protein or something that promotes fiber self-association, what will happen is that larger assemblies will form, they will be heavier, and they will tend to sediment at the bottom of the cube upon centrifugation. And so that's exactly what happened when I started to add HP1 protein. You can see on the x-axis, you have increasing concentrations of HP1. On the y-axis, you have the amount of chromatin soluble in solution. And so when you start adding HP1, the chromatin in solution tend to decrease. And so this indicates that the HP1 proteins are really promoting fiber-fiber self-association. So that was the time when we started to think about phase separation, and HP1 phase separation was discovered. And so that was why I decided to take this chromatin HP1 assemblies and look under the microscope. And that's when I observed the formation of this liquid droplet of condensate. So you have here under this condition HP1 alone in a test tube, which is in solution. Here you have chromatin. But when you start to put together chromatin HP1, you start to see the appearance of this condensate. And the formation of this condensate is really correlated with the pelleting that was detected in the pelleting assay. So this really indicated, this was the first evidence that HP1 was mediating phase separation of chromatin. What I also did was to label chromatin. This is a Sci5 label instance. And so what I realized is that chromatin is really highly enriched inside this condensate, indicating that HP1 is really concentrating chromatin in there. So this was really the first evidence that compaction of chromatin mediated by HP1 at least was coupled to its phase separation. So now the question is, what is the role of the reshaping of the nucleosome core in this context? And what I did was to repeat the same assay, but this time I prevent the nucleosome conformational change by locking the nucleosome core with site-specific disulfide bond. If now you use this chromatin fiber where the nucleosome cannot breathe anymore, we can see a defect in the pelleting assay mediated by HP1 shown here in this green data set. As well, we see corresponding defects in the formation of this condensate. So this was exciting because it meant that the conformational change in the nucleosome core was important for chromatin compaction, but also meant to me that it was the behavior of the nucleosome itself, of the single monomer, could really influence and regulate the behavior and the folding of an entire chromatin fiber in the mesoscale. I'm not going to go into the detail of the in vivo data, but what I want to tell you, just one highlight, is that if you look in cells, and I told you at the beginning, HP1 formed these very bright foci, which are highly dynamic. And then if we start to introduce mutation in HP1 that either abrogate phase separation in a test tube or alter phase separation properties, we start to see the corresponding changes also in cell in terms of formation of the foci, but we also start to see the effects in gene silencing. So this indicates that the interaction between chromatin and HP1, which mediate phase separation in the test tube, are also relevant for proper heterochromatin formation and function inside the cell. Okay, so what we learn in terms of HP1 and heterochromatin using this combination of biophysical method that I told you about. So we knew from previous work that HP1 proteins bind chromatin containing methylation of lysine 9 of histone H3, and they have the ability to oligomerize and potentially bridge across the nucleosome. But what we have learned now is that also by binding the nucleosome, HP1 induces a conformational change. And this conformational change in the nucleosome core opens up the nucleosome and expose regions that are normally buried inside the hydrophobic core of the nucleosome. And this opening up of the nucleosome is actually important to drive chromatin compaction and the formation of this condensate. So what we propose is that the exposure of these hydrophobic regions in the nucleosome core can now be available on the surface to template nucleosome-nucleosome interactions. And these interaction, dynamics and weak transient, are the kind of interaction that can drive compaction of chromatin and phase separation. So you can kind of think of the exposure of this hydrophobic region of the nucleosome as just a way to increase the opportunities for internucleosome interaction. And now this increased accessibility at the level of one single nucleosome start to make sense, because we realize that by increasing the accessibility, the exposure of one single nucleosome, we're actually creating a chromatin fiber that is overall more compacted. So I'd like now to zoom out a little bit and get out of the HP1 heterochromatin context to think more broadly how this observation in the context of heterochromatin can help us to rethink some of the long-standing concept of chromatin organization. And so that's how, based on that, I really propose what I call a liquid-like model for chromatin organization that kind of revise and introduce some new concept compared to the model, the hierarchical model I showed you at the beginning. So in this model, there is, of course, nucleosome conformational dynamics. You probably can see inside this condensate, nucleosomes have the form structure. And I told you today that these conformational changes in the nucleosome are actually important to regulate chromatin compaction. But we can really start to think of nucleosome conformational dynamics as a potential new layer of chromatin regulation that can regulate many different aspects of chromatin biology. And so you can start to think of this as, for example, an extra a new code, a nucleosome conformational code that can function in addition, for example, to post-translational modification and coordinate and co-regulate chromatin structure and function. And of course, an aspect I'm particularly interested in my lab is to investigate the extent of these conformational changes and the function that they might have in cells. The second concept, of course, and this is the relevant for the group of people that I'm talking with today, is the concept of phase separation as the possible principle that can contribute to chromatin functional organization. And so, and I still think there is a lot to need to be done to address, to study in a quantitative way phase separation in the nuclear context and understand what's the role and whether it regulates and how it regulates transcription and many other processes in the nucleus. But even from a very early days in terms of understanding this process, what I think is extremely exciting is just the perspective that phase separation provides in this context. And this perspective is the fact that chromatin can still be organized in cell in absence of this highly ordered structure that I talked about at the beginning. So we don't really see a 30 nanometer fiber in cell, but this doesn't mean that chromatin is a messy ball of spaghetti. So we can still get some level of organization, but through structure and interaction that are much more dynamics than we previously thought. And we can start to think of a group of interactions that are weak and transient and can regulate chromatin function and organization on the mesoscale. So this means really start to think about chromatin as a much more fluid polymer and really investigate a different type of interaction in terms of weak and transient interactions. So I also want to spend a couple of minutes to think about chromatin itself. So we tend to think of chromatin as a really passive occupant that sits there and doesn't do anything. We used to think of transcription factor, HP1 proteins coming on chromatin, doing their job, and just chromatin being in some kind of inert. But what this conformational change in the nucleosome is highlighting is that actually chromatin contributes to multivalency. And that also means that the intrinsic property of a chromatin can really fine-tune and regulate the process of chromatin phase separation or mesoscale organization. So chromatin composition can self-regulate its own organization. And this has been also supported by some other recent data. This is a work that has been published recently from Mike Rosen's lab in the context of chromatin. What they did was to start to look at a series of chromatin intrinsic properties that have been shown to be important to regulate chromatin compaction. And so what they were able to show is that all these elements were also important in regulating chromatin phase separation in the test tube. So this is an example. You have the formation of chromatin condensate in a test tubes when you have normal nucleosome. If you chop out the nucleosome tail, you really lose the formation of this condensate. This is telling us that the tails of nucleosome are important. able to show the post-translational modifications on the tails of the nucleosome, specifically acetylation, fine-tune the process, as well as the spacing of nucleosomes, how far from each other the nucleosomes are in a chromatin fiber. So this is telling us that there are really intrinsic property of chromatin, including nucleosome core plasticity, that can fine-tune and regulate chromatin mesoscale organization, but also is telling us that chromatin is actively participating and providing multivalency. And I also want to spend a couple of minutes to think about chromatin as a polymer and thinking about the phase separation in the context of very long polymer. And so there is work here again from Mike Proson's lab that showed that the length of the chromatin polymer test tube can fine tune and change phase separation. So you need, of course, a minimal length of monomers of nucleosome to be able to observe enough multivalency to see phase separation. But there's also been a recent report that showed how the length of the DNA can also regulate the properties of the condensate observed, in this case, in the context of HP1. So here you have condensates that are formed in the presence of 2.7 kb piece of DNA, and here you have the same experiment performed in the presence of 9 kb piece of DNA. So you can see that the length of the DNA is really changing the formation and the properties of the condensate. And this work really started to dissect what were the difference in terms of dynamics and viscoelastic property of this condensate. So this is really making us think that the length of this polymer is, not surprisingly, is really regulating. It has a role in fine-tuning phase separation. But I think it's interesting to start to think how the chromatin long polymer in the cell is actually regulated, and how we can have different condensate or different type of chromatin formed within the same long polymer. So I think this is an exciting venue for research, and just think about this for what that means in terms of chromatin organization in the nucleus of a cell. Okay, just to summarize, an important message that I want to try to put out there today is really to start to think chromatin in a different way, and not just think about chromatin as a passive occupant of HP1 droplets or transcription factor, but really something that contribute to the self-organization and 3D organization. I mentioned that it's really interesting to start to think how a monomer, the nucleosome within the polymer, within the chromatin fiber, can contribute to the organization of the entire polymer, of the entire chromatin fiber, and start to think how the different flavors of nucleosome can contribute and add extra layer of regulation in this process. I talked about the conformational change in the nucleosome itself, so atomic scale conformational changes, and how much this is linked to the mesoscale organization of chromatin. And so I think it's really interesting in the future, and it has been started to show in different example of condensate, start to think about conformational switches and how understanding conformational changes in protein and in complex can help us understand the regulation and the formation of this mesoscale assembly. In the context of chromatin, it's also very interesting to notice that there are both different types of interaction that are involved in the formation of the condensates. There are electrostatic interaction, which involves interaction with the DNA, but it also will start to have hydrophobic interaction that involve the hydrophobic core of the nucleosome. So I think it's the chromatin from this perspective is a very interesting polymer when we start to think about the respective role in hydrophobic and electrostatic interactions in regulating phase separation. And then of course, in the context of HP1 heterochromatin, we start to see that we have both homotypic and heterotypic interactions. So we have interaction between HP1 proteins, so HP1 interactions. We have interaction between nucleosome, internucleosome interactions, and also we have interaction between HP1 and the chromatin. And so I think this is just highlighting the complexity of the flavors of phase separation can have in the context of a complex system such as chromatin and chromatin binding factors. And to end this talk, I just want to make a remark about the HP1 heterochromatin system I talked about today, and the fact that if we start to look closely, we start to see that all the ingredients that are known to be involved and be important for phase separation are really part of the mix. So we have the role of disordered protein regions, so that we know are often involved in phase separation processes. And you can think of the histone tail, that histone tails are disordered flexible regions, HP1 protein as a multiple region that are disordered and structure. There is macromolecular crowding, and this is an aspect that still needs to be addressed, but the nucleus is actually a very crowded environment. And then there is the role of weak multivalent interactions that are known to drive the formation of these condensates. And then there is a polymer. You can think of chromatin, the very long polymer, and also HP1 protein can oligomerize and form these long chains. So if we look at all the ingredients in the context of HP1 heterochromatin, I think it's not so surprising that we observe phase separation. But the exciting and the challenge for the future is really try to understand how this physical chemical principle in the context of chromatin can regulate its properties and the function, and really trying to understand, learn from these different fields to be able to better understand genome packaging and genome regulation. Okay, I'm gonna end here and just spend a few seconds to thank my supervisor, Gideon Arliker and John Gross at UCSF. Those were my mentor during my postdoc, my collaborators, and this is us, me, the first members of my lab. I'm packing boxes and trying to set up the lab. And so if you wanna help us open boxes, you can reach out to me and I'll be happy to chat more also about science. I'm happy now to take any question. I hope that will be a fun discussion.
Yeah, thank you, Serena. And there are lots and lots of questions coming in. So really stimulating talk. Everybody loved it.
Thanks.
Maybe we could do this in all kinds of different ways. I would say maybe let's start with Phil's question. Towards the end of the chat window, Phil's asking about evidence for the dynamic nucleosome conformation isn't also present in euchromatin.
This is a good question. Yeah, I do think of, there is no evidence in the sense that we don't really have enough data. It's particularly challenging to look at nucleosome conformational changes. And so I think it's the last 2-3 years that people are starting to look at it. And my suspicion is that there are nucleosome conformational changes in multiple chromatin contexts. And one question is, do you have specific conformation in heterochromatin versus euchromatin, active versus repressed regions? And this is one possibility, but we need to do more. I really think of nucleosome as something very squishy that tends to be highly dynamic and sample multiple conformation. And then one possibility is that any factor that engages the nucleosome core can bias this conformation and stabilize one of these conformations, or you can think that the binding of this factor can also push and drive a very different type of conformation. So those are all questions that we don't know. With the technique that I mentioned, HDX and NMR, we are able to see things that are moving, but we don't really have a snapshot, a picture of how this distorted structure looked like. And this is some of the question that I'm interested. And so I'm developing tools such as, for example, nanobodies to be able to trap and study some of these conformations. But the most recent cryo-EM structure of nucleosome bound to factors are showing that we're starting to see, even with cryo, some conformational changes. And so we have missed this partially because technically it's been challenging and we didn't know that something was going on there, but also because people tend to push for a high resolution, and so they discard everything that deviate from the most stable conformation. So I suspect that more and more will be coming out in the next couple of years. But for now, we just have hypotheses, and I think I suspect that we'll see more and try to determine more what has to do with euchromatin versus heterochromatin.
Yeah, and actually that all makes total sense. And it also leads to another question that Rosanna asked, where she's also asking about, you can see it in the chat window, this idea of nucleosome breathing and, other protein changes to the way the octamer is assembled. So again, it's all part and parcel of this same need to really be thinking about the subtleties of the dynamic motions of these.
Yeah, I'm not sure if I understood the question right. If in terms of thinking, the process of assembly and disassembly of nucleosome on chromatin, we know that our intermediates, it has to do with how our nucleosome are put on chromatin and how they're moved around. For example, there was another lab that showed that if you want to slide nucleosome along chromatin in a DP-dependent manner, you need to also distort, have a flexible nucleosome core. So I believe this is a process that has to do with intrinsic property of chromatin that can be exploited in different contexts, as well as assembly. But I was particularly surprised in the context of HP1 protein is that there is no there is energy, binding energy involved, but there is no real energy that I was thinking of involved. In the context of chromatin remodeling, when we saw that the remodeler was pushing the nucleosome along DNA using ATP, we saw distortion there, but we thought, it's a motor, is pushing nucleosome around, that's why you need distortion. But when we think about HP1 proteins just sitting there, the idea that I had that they were not going to induce such a huge conformational change, which suggests that many of these changes might really occur broadly in chromatin processes. It's just that we haven't thought about that, and also haven't been able to see that technically.
Mm-hmm. That's great. There's another, there's so many questions. Another one that I think is interesting comes from Bede, who's asking, and I maybe could ask Bede to unmute to build on his question, but he's asking about the acidic patch on the nucleosome and whether that's a docking site, whether that can be interacting with your chromatin, and then whether you see evidence from HDX or other methods.
Yeah, so this is a good question. So the acidic patch is this region at the interface on the top of the nucleosome formed by histone H2A, H2B, and it's basically the only acidic site in the whole nucleosome, and has been shown to being the anchor site for many transcription factor or chromatin binding factor that interact with chromatin. So I definitely think that the acidic patch has to do with this process. There's some work that's been done by Mike Rosen's lab in this context. And also because the old chromatin compaction model based on the 30 nanometer fiber in a test tube were showing interaction between one tail of the nucleosome H4 and this acidic patch. And those were kind of interaction will mediate packing. So I do think that this interaction are likely involved also inside a condensate. And this is one of the things I want to do in my lab is to address better what are the interaction involved. And this will be one of this. One of them. One interesting aspect in terms of HP1 is that HP1 does not seem to use the acidic patch. I did a bunch of experiment and look at the acidic patch because that was the suspect that I thought of when I started to think of interaction between HP1 and nucleosome. And surprisingly, HP1 does not use the acidic patch. It doesn't care at all. So for me, it is interesting because you can start to think that maybe HP1 binds reshape the nucleosome and deform the acidic patch in a way that can promote interaction with one factor versus another on chromatin. So you can start to think about the layer of regulation that can be down the hill. So yeah, that's exciting. Yeah.
That's great.
Steering via condensation, basically.
Yeah.
Yeah, cool. Thank you.
Yeah, so Jeremy, you had some questions, if you want to jump in. I think maybe you've been unmuted. Or Suzanne.
Yeah. Yeah, okay. So my question was, let's see if I can remember. So the claim was that that you get the binding on there and it creates some conformational change inside the nucleosome, and that it reveals new interactions that allow them to come together. And I was just thinking that an alternative model is just that the conformational change in the nucleosome just allows a higher affinity of the HP1, and so what you're seeing is just an enhancement of the HP1 binding, effectively just a higher equilibrium constant there. Does that explain the data, or can you rule that out some other way?
Let me see if I reframe the question. Let me know if I get it right. So there are basically what I was trying to do. You have the two sides of the thermodynamic box, right? There is one way where you can think that nucleosome is just highly dynamic in sampling this conformation, and that HP1 recognize one of these alternative state, bind that, and stabilize that.
Right. Yeah.
The
Yeah.
other option is that the nucleosome is sitting there, and then the binding of HP1 induce such a conformational change.
Yeah.
So the outcome of the thermodynamics of the two sides of this thermodynamic box is the same, where you have an opening up and a stabilization of more open nucleosome state. I can't rule out which one of the two pathway
Okay.
are in place. I don't.
But you seem to be pushing a third model there, where that once you rearrange the nucleosome, that exposes some new interaction sites, and those facilitate the compaction.
Yeah, so that is what happened after this conformational change has happened, right? In the context, when you have a chromatin fiber present there, when those regions are exposed, then now can be available for the interaction with chromatin, or potentially other factors that are interacting with chromatin itself. So those are like the downhill effect of this stabilization of conformational change that is going on. I also want to
so how do you know that it's not just the HP1 effect that explains everything?
So there are, if you prevent, I can tell you if I get it right, that this conformational change is HP1 specific. Is that what you're trying to say?
Well, I'm just saying I'm just saying, how do you know that it's not just enhanced binding of HP1 that's facilitating the compaction? How do you know that there's other stuff that's coming in? I mean, I believe that there has to be some other stuff that comes and sticks to it, but how do you know that that other stuff that sticks to the exposed parts of the nucleosome is actually facilitating the compaction?
Yeah, so to really understand what those hydrophobic region exposed are doing, we need to do extra experiment to figure out what are they interacting with. To say something about what you say that maybe can help understand is that if you do induce compaction of chromatin in a different way, not using HP1 but using hydrophobic isol and divalent salt, and you prevent this conformational changes, you also start to see the effects in the compaction. And so what I'm thinking is that what HP1 is doing is really promoting it, taking advantage of an intrinsic property of chromatins and just facilitating the interactions between nucleosomes.
Okay. All right. Thank you.
But I also want to make clear that I don't think of nucleosome as something sitting there and being static in one state. I think of nucleosome as highly dynamic itself. And then the binding, we need to think that whenever the nucleosome is interacting with a protein, it's not just simply sitting there and being inert, but when you engage with the nucleosome, there is a response and a conformational change that occurs there. And this is not surprising if you think about any protein complex or group of protein coming together. Just for some reason, chromatin people assumed that there was nothing interesting going on that portion of the nucleosome.
That's great. So there was an interesting question from Sue Swally, if she's still on.
Yeah, I'm here, Mark.
Greg, why don't you go ahead?
Yeah, fantastic talk. I was really struck by sort of that canonical image of chromatin, and then when you actually looked in vivo in the cell, how different it actually looked. And in the talk where you were showing Rosen's work where they cut the tails off and that affected the droplets and the ability to form droplets, what does that actually look like in the cell? Do you see a difference between in the cell when you do those sorts of experiments?
So I don't think that the chopping off of the tail has been done in cells. In general, manipulating histones in cell is very challenging technically because you have multiple copies. In some cases, you have 10, 20 copies of each single histone. So doing this kind of modification is really challenging in cell. If I recall it well, the only experiment that Mike Rosen did in cells to test some of the in vitro regulation processes in terms of phase separation was to play with the acetylation of the histone tail. So acetylation is a charge rest put there on the histone tail has been shown to unpack chromatin, opening up, preventing the interaction between the acidic patch and the tail that we talked about before. And so what it was able to do was to induce acetylation and acetylated chromatin into cell and see that would form condensate when it's puncta, when it's not acetylated, and when you introduce acetylation, you dissolve this condensate. So if I recall it well, that's the only interactions involving chromatin or regulation that has been fully explored in vivo. And I don't think anyone has ever chopped the tail of the nucleosome to look in cell. Yeah.
Great. And now we're going to run out of time, although Serena, you said you can hang around for a bit. Is that right?
Yes, yes. And the light is on in San Francisco, so now it's daylight, so it's much easier for me to be awake and chat more with you.
You mean it's not sunset over the Golden Gate Bridge?
No, it's not. But before was really night. Yes, yes.
So maybe we'll go with one more question before we, I know a lot of folks will have to leave at the top of the hour. So Charlotte had a really interesting question about the idea of aging or hardening. So Charlotte, are you unmuted?
Yeah, I think so. Can you hear me?
Yes.
Okay, great. Thanks for a really exciting talk, Serena. My question is that so many other condensates have been demonstrated that if you let them just sort of sit around for a while, they start to age and harden. And I was wondering if heterochromatin might also undergo this process, and if not, what's helping to keep it dynamic so that I can then, switch between being open and closed.
Yeah, so the hardening phenomenon in test tube is also observed in the context of HP1 and chromatin. So if you start to wait longer, the fusion events will just be harder, and the droplets will not be able to relax, as we see in this video that I show you today. So the aging and the hardening process seems to also occur in the context of HP1 chromatin. I haven't really explored that further to understand what that means. I agree that the question is, so how is this regulated in the cell? Why don't we got this hard body or gel or everything forming all the time in the cells? And this is, I think it's an opportunity to remind that heterochromatin, everything in the cell is more complex, but specifically heterochromatin is a very complex platform. And HP1 protein not only binds chromatin, but it has also a domain that is known to recruit specific factors. It's really a hub for recruiters of other proteins on chromatin. So this means that what we observe in a test tube with simple composition, just HP1 and chromatin, could be very different when we are in contact with different proteins. So all these proteins can contribute to maintain a dynamic property of the interaction on the condensate in cells. So it does happen in our test tubes. I don't know in vivo, but again, a lot of components are dynamically regulated in terms of chromatin itself, heterochromatin itself. And I want to use this as an opportunity to just share with you the way I think about HP1 and phase separation. And this is not really supported by data, but it's just the way I think about it. I don't think of like phase separation and phase transitions of different material states to happen all the time when you add HP1 into the cell or a chromatin. I really think that of an extra layer of regulation that can be in place and can be really dynamically fine-tuned and modulated in the cell. And so the material state and likely is going to be coupled to the composition and many other the rest of the complex environment that you have in the nucleus and within heterochromatin.
Yeah, that's great. So let's go to Tanzim next, if he's still on. If not, let's go to Oscar. We have lost Oscar.
Hi, Serena.
There you go.
Yeah, very nice talk. So I wonder if you tried this reshaping on different type of heterochromatin proteins, such as MECP2, or even the mammalian version of H1 alpha?
No, this is a good question. And, I think that now we have this setup, which is a combination of NMR, HDX, and cross-linking mass spec. That's the next thing, right? Trying to figure out how the different how the different factors can affect the nucleosome itself, and can we find a pattern? Can we find specific changes associated with activation repression, or can we identify hotspot region within the nucleosome that are subjected do this kind of changes? So this hasn't been done, and this is mainly a technical reasons. Performing the experiment that I told you about is actually very challenging. Just to give you an idea, to perform the NMR experiment setup, it will take 6 to 8 months to make a sample to be able to do one NMR experiment. And it's kind of similar for doing HDX. So those are not high throughput experiment that you can perform and use different proteins so easily, but it can be done using with any protein you can think of, but you'll need to really understand the system also very well. So in the context of the experiment I've done, I used the yeast version of HP1. I had a very good understanding of all the moving parts of the system, let's say. So I was able to perform this very difficult biophysical experiment that I mentioned, that I talked about.
That's great. So yeah, I think it will be very exciting to also know about how histone H1 influenced this, reshaping the chromatin by H1 alpha or maybe other heterochromatin proteins.
Yeah, there is some data out there in the context of H1. So H1 is this linker histone that sits at the entry exit of the DNA, and it's like putting a color on the nucleosome. It's like it's known to promote compaction. So we know from Mike Rosen's lab that the presence of the linker histone H1 promotes compaction and phase separation. And there's been past work of NMR in the presence of the histone H1 that they did not detect any conformational change driven by HP1 in the nucleosome itself. So it doesn't seem that HP1 seems to induce a conformational change in one nucleosome itself. And so the conformational change may be a consequence of stacking of nucleosome, which is promoted by H1. So this made me think that in order to see a conformational change in the nucleosome induced by a chromatin factor, my way of thinking about it is that the factor needs to engage the nucleosome core to really sit, not acting just from the tail, but really engaging the folded core of the nucleosome. And the linker histone is very small. It's just sitting at the not really at the exit of the DNA. It's really sitting on the DNA. So the way I think about it is not really contributing enough in terms of binding to push to change the conformation of the folded core.
Great. So Tanzim is next.
Yeah. Hi, very nice talk. I was wondering what would be the dynamics of HP1 mediated chromatin compaction would be depending on the cell cycle of the cell.
Are you talking about dynamics in terms of conformational change of HP1 itself, or you're talking about binding release from chromatin? Which scale of dynamics?
I was just wondering that it has been known that H3K9 trimethylation can change depending on the cell cycle of the cell. And what I understood from your talk is the compaction of the chromatin by HP1 is very much dependent on H3K9 trimethylation. So I was wondering what would be the dynamics in that perspective.
Yeah, so this is basically discussing how is HKK9 deposited throughout the life of the cell, the cell cycle, as you're mentioning, after replication. So this hasn't been explored. And just to add an extra, what you just said, another key regulator of HP1 phase separation and HP1 in general is the phosphorylation of HP1 itself, which seems to be cell cycle regulated. And so, and this conformational change in HP1 driven by phosphorylation, controlled by phosphorylation is also affecting interaction with chromatin. So those are very good questions, and exploring I think this is more broader than phase separation, is really exploring the assembly and disassembly of heterochromatin in cell. And there is a lot of work out there on this, and I'm not expert on that, but I think it's a very good question, and it will be fun to think more about that from the perspective of the knowledge that we have coming from phase separation.
Okay, thank you.
Great. Maybe we'll go to Rosanna. Thanks.
Hi. Yeah, so I already asked my question, but I was saying to Jeremy regarding his point about how do you know that is the nucleosome deformation that actually promotes liquid-liquid phase separation and not enhanced binding of HP1, for instance. We have done some simulations in which we have only used chromatin, and we do observe that similarly to what you just described, just nucleosome breathing enhances the multivalency of the nucleosome. So it promotes these enhanced liquid network connectivity and also enhanced flexibility of the chromatin system. So it's exactly what you described that we have tested just with modern.
Yeah, and I think I saw that paper, read that paper carefully, and this was exciting. And that's, I don't think is, I mean, the changes that we see are somehow specific for HP1. There is some level of specificity. I've tried a few factors in the test tubes and see if they care about, cross-linking of nucleosome and locking the nucleosome, and I didn't see any changes, but I haven't looked broadly to all the transcription factor you can think of on one side. And on the other side, there is another thing that made me think about specificity is that the conformational change in the core of the nucleosome the context of a methylated nucleosome. If you remove the metal part of nucleosome, and HP1 still binds, but it's not able to induce that conformational change. So I think there is some level of specificity that is regulated by HP1, but I don't think it's an exclusive thing that's happened in the context of HP1. And in multiple aspects, even intrinsic, or you can think about the histone variants, right? And changing the composition of the chromatin itself, how much can just, it will be fun to put in the modeling that you did, the histone variants and see how that would affect the process. Yeah.
Yes, or also like DNA sequence variations as well. And yes, thank you.
And then, all my work has been done in the context of this optimized, strong sequence positioning. So you can think that if you have a less strong, the dynamics and the ability of the nucleosome to breathe will be increased as well. And the spacing between nucleosome and a chromatin fiber seems to be determinant for microsomes work. And it was fun to see that the spacing that seems to correlate in vivo with euchromatin, it was a spacing that was not favoring phase separation of the test tube, versus spacing of nucleosome that is tend to be found in heterochromatin or compacted region is a spacing that favors chromatin phase separation. So interesting.
Thank you.
Thanks.
So let's go next to David, and then Gaurav, and then Patrick. So David, you're up next.
We can't hear you. I think you're muted or something else.
Yeah, if David was on a second ago. Oh, there
Yeah.
you are. Great, David.
Okay.
Yes.
Yeah, sorry. I was just wondering if there's quantitative data in terms of dissociation constants between nucleosomes, so the internucleosome affinity for different histone variants, or also maybe in presence of HP1.
I don't think so. I'm not aware of any of this information out there. This is a good point. Yeah.
Thanks.
So Gaurav, you're up.
Hi, thanks for a great talk. It was really amazing. So I have a question about, so like the way I'm thinking about chromatin is as this polymer. And when you show these phase separated condensed states, the way I think is like sort of one length of the polymer has been phase separated by these HP1 molecules. So I'm wondering, like, what is determining that size of the length of the chromatin which gets sort of phase separated, if you will?
It's a good question. I think this was the kind of thought that I was trying to provoke when I mentioned at the end of my talk the initial data that we have in the context of HP1 and length of DNA and the number of nucleosome. I think this is a good question. The chromatin, it's very long in the cells, and so how that gets organized, and if you have subregulation, what is the boundary that create all that regulate the process? When do you stop phase separation? How do you determine which nucleosome get included and which one get excluded? So those, I don't have an answer for that, but I think it's fun to start to think about that and connect that with what we know. I think just thinking about boundaries and transition from one chromatin, not phase state, but just chromatin state, as we used to think in the past, like kind of type of chromatin. It's fun it's not well understood, what is the boundary that prevent heterochromatin to spread? There is a lot of work that has been looking at what prevent heterochromatin to spread where it's not supposed to go. And so I think it'd be fun to start to bring that knowledge in the context of phase separation. But yeah, I don't have an answer for that, but I think it's an exciting something exciting to think about for the future.
Great. So
Thanks.
let's go to Patrick, and then I think we'll close with a question from Alan, and then we'll have to cut it off. So Patrick, you're next.
Cool. Thank you. Thanks, Serena. Really, really interesting talk that brought a lot of different details of a very complicated problem to bear. I was particularly interested in the NMR data that you showed. I identified lots of specific, but also distant, spatially distant residues that showed conformational changes on timescales you were probing. Do you think that those are cooperative in some sense, or should we think of them as all independent? I guess it's coming from a standpoint of, if you think about the energetics involved in the conformational changes, how much energy is required and how much can you actually be liberated from HP1 binding?
Yeah, that's a good question. I don't think I have any experimental data to answer this. So just in a speculative way, I tend to think of this as a cooperative process. And the reason is that, I haven't done NMR experiments with different mutants and breaking specific interaction between the nucleosome and HP1, so I can tell you that. But if we do easier experiment, when we look at affinity or we look at phase separation or compaction, if you start to break some of these interactions, you start to see defects. And so, for example, if you and there are multiple domains of HP1 that engage different regions of the nucleosome. And so when you interfere with some of that, for example, you start to see defects in phase separation or defects in binding. And so I haven't tested, there is an experiment I could do to test this. So I tend to think of this as a cooperative process, because for example, I was telling you, if you remove the methylation of the instance tail, which you're basically just losing one interaction, right, from the tail to one specific domain of HP1, you lose the importance of the conformational change in the process, because the mutation, the abrogation, blocking the conformational change does not cause any defects in the process. So I think there are few of these interactions seems to be cooperative. I think actually now that I think about it, it seems to be cooperative, but I tend to think about that in a cooperative way, at least in the context of HP1, and the fact that you really need to hold nucleosomes from different parts.
So I think, to me, that suggests then that you have a relatively small number of adapted or non-canonical states of the chromatin then. Because if you imagine that all of them could be happening independently, then you have lots of different combinations of conformational changes. But if it's cooperative, then you really have sort of a switch between two states in the simplest limit.
Okay.
And I think that has implications for the cooperativity.
Or some discrete number of states, but not a huge number. Yeah.
Absolutely. I mean, it's a continuum. Absolutely. But yeah. I think it's helpful to know which end of that you're on, Avogadro's number or accountable on my hand number.
Yeah, I see what you're saying. Yeah, I didn't know that.
Cool.
Yeah, that's interesting. Yeah.
Thanks so much.
And so one of we can do now also NMR experiment, CPMG experiment on the nucleosome itself alone in absence of HP1. That's easier, where we can start to really look, and determine some of these discrete state and the dynamic range of that conformational changes. And these are some of the experiment I'm planning to do if I find a person crazy enough to do NMR on nucleosome.
So we'll go to Alan.
Congrats on your appointment. Really nice work.
Thank you.
I can't recall the phenotype from like the HP1 alpha beta gamma knockout cells, but in the CYP39H12 knockout cells where you lose H3K9 trimethylation and heterochromatin, you don't recruit HP1 anymore. You don't see a chromatin decompaction phenotype as far as I know. So I'm just trying to reconcile what you're seeing in vivo with reducing chromatin in vitro with chromatin compaction and what actually happens in a living cell.
I thought that when you really remove all the HP1 proteins, you lose completely gene silencing and you have the repression, but you also lose compaction. You don't lose it? You say you're not losing compaction when you remove all the HP1s?
Well, I mean, if you take a mouse cell, chromocenters usually persist, and there's actually only very few proteins that when removed from the cells cause those structures to actually disperse, in mice at least. And just the overall idea about how heterochromatin organization varies across species as well is difficult to reconcile with just HP1 behavior in general, but just because HP1 itself is so well conserved. But
yeah, it is well conserved, but at the same time, really small changes seems to have huge impact in the way HP1 interact with nucleosome and induce phase separation. And so, for example, there's been work that compare HP1 alpha, beta, gamma, human from in different different sequences of the domain that have small differences that seems to really affect the interaction with DNA, their phase separation ability, and so on. So I think the small changes might have large impact. What we think as small changes might actually have large impacts on the way they interact on chromatin and fine-tune phase separation. So I don't think I don't know in vivo how to reconcile the data. And I don't think I know well enough the data you're talking about to comment on that. But I just think that we should not underestimate small changes in sequences and differences. And this is true for HP1, but also for histons, for example. Sometimes you have one point mutation, which seems to dramatically affect completely change the function of chromatin with no substitution. So especially for processes that are so linked to conformational dynamics, and HP1 has huge conformational dynamic in itself, I think small changes can actually dramatically change the type of conformational dynamics on the time scale of these dynamics.
Thanks.
Well, this has been wonderful. And the fact that, you know, so many people have hung around for an extra 20 minutes speaks to how interesting of a topic this is. So Serena, thanks again from all of us. Wonderful lecture.
Thank you so much for inviting me and for the fun discussion. It was worth to wake up so early.
Good. Glad you feel that way. Thanks you. And thanks to everybody for joining.
Thank you so much. Have a good day.
Thank you. Bye-bye.